All right, let's dive into a real scientific detective story. It's all about a revolutionary idea that completely shattered our understanding of one of the most fundamental things in life, the gene. For the first half of the 20th century, scientists were pretty sure they had the gene figured out. It was like the atom of heredity, the smallest possible piece, indivisible and absolute. But what if all of that was completely wrong? What if you could actually crack that atom open and find a whole universe inside? To really get why this was such a big deal, we have to go back in time. We need to understand the old dogma. So, what did scientists think a gene was back then?
Well, the leading theory was actually pretty simple and kinda elegant. Picture a chromosome as a long string, and threaded onto that string are these little individual beads. Each bead was a gene, a single, unbreakable unit responsible for one specific thing. You couldn't split a bead, and you sure couldn't look inside it.
And this wasn't just some casual idea, okay? This was the central dogma. This beads on a string model basically said that genetic shuffling or recombination could happen between the beads, but never ever within a single bead. The gene was a fundamental particle. Case closed, or so they thought. But scientists are detectives at heart, right? And they started finding these little clues that just didn't quite fit the model. And the first big tool they developed to investigate this was something called the complementation test. Okay, so imagine you've got two flies, and neither of them can fly. They have the exact same problem, what we call the same phenotype. But is the genetic mistake in the exact same spot for both of them? Is the same little bead on the string broken in both flies? That was a huge question. How could you possibly know? Well, the solution was this really clever idea called the complementation test. You might also hear it called a cis-trans test. Its job is actually pretty simple. It's there to figure out if two mutations are in the same gene or in different ones. It's like the first step in our investigation. Are we dealing with one broken part or two totally different broken parts? And here's the beautiful logic behind it. On the left, let's say you cross two mutants, but their mutations are in different genes. Well, each parent gives the offspring a working copy of the gene the other one is missing. They basically cover for each other. They complement each other. And poof, you get a normal, wild-type kid. But on the right, if the mutations are in the exact same gene, well, there's no working copy to go around. The offspring is still a mutant. It's a simple idea, but absolutely brilliant. So complementation tells us if we're in the same ballpark, the same gene. But the second huge clue allowed scientists to start measuring the actual distance between those genes on the chromosome. This next clue comes from a totally natural process called recombination. During meiosis, when sperm and eggs are being made, chromosomes pair up and literally swap pieces with each other. This genetic shuffling is the reason why siblings aren't identical. It's constantly creating new combinations of traits. And here's a look at how it works. See, the F1 generation has one chromosome with the alleles A and B, and another with lowercase a and b. During gamete formation, recombination can swap them, creating brand new combos like A with B and A with B. Those are what we call the recombinant gametes. Now, here is the absolute key point. The chance of a swap happening between any two genes depends entirely on how far apart they are. Think about it. If two beads are really far apart on the string, there's a ton of room for a crossover to happen between them. But if they're sitting right next to each other, a crossover is going to be way more rare. All of a sudden, recombination frequency became a kind of genetic ruler. And using this exact principle, pioneers like Thomas Hunt Morgan created the very first genetic maps, like this one here for the fruit fly. This was monumental. They could actually chart the order of genes and their relative distances. But, and this is a big but, they were still treating genes as single points, as those indivisible little beads. And this brings us to our hero, the detective who took all these clues, put them together, and just cracked this whole case wide open, Seymour Benzer. So Benzer asked a question that was almost heresy at the time. Everyone was busy mapping the distance between the beads on the string. Benzer looked at this and wondered, hey, could we use recombination not as a telescope, but as a microscope? Could we actually map the distance between different breaks inside a single bead? To do that, he needed a system where he could look at millions or even billions of genetic events all at once. He found the perfect tool for the job, and the T4 bacteriophage. It's this tiny virus that infects E. coli bacteria.
and he focused all his attention on a specific region called RII, which everyone at the time considered to be a single gene. His method was just a 4-step masterpiece of an experiment. First, he made this huge library of mutant viruses, all with a broken RII gene. Second, he used complementation tests to sort them all into 2 distinct groups, which he called cistron A and cistron B. And then, here comes the masterstroke. He took mutants from the same cistron, mutants that failed to complement each other, remember, and crossed them. He was hunting for incredibly rare recombination events within that single functional unit.
I mean, you have to understand, this idea was completely radical. Genes were supposed to be indivisible. End of story. His own colleague, the famous geneticist Max Delbruck, thought the idea was just outrageous. It went against everything they thought they knew.
But Benzer proved them all wrong. His system was so unbelievably powerful that he could detect recombination frequencies as low as 0.02%. So what does that number even mean? It means he was mapping mutations that were just 1 or 2 base pairs apart. He wasn't just looking inside the bead anymore. He was practically reading the inscriptions on it.
So yeah, Benzer's experiment changed biology forever. It's time for the big reveal. Let's look at the new picture of the gene that emerged from all this. This slide just says it all, doesn't it? We went from the old view, the gene is a fundamental, indivisible bead, to the modern view. A gene is a physical length of DNA, it's a linear sequence of nucleotides, it has a beginning, a middle, and an end, and it can absolutely be broken and recombined right within its own borders. The bead was shattered.
And this table gives us the perfect summary of the 2 tools in our detective kit. Complementation asks a simple yes or no question. Are we broken in the same place? Recombination is our ruler. It answers the question, okay, but how far apart are the breaks? Benzer was a genius because he used both of these tools together to create a high-resolution map of a single gene.
So why does all of this matter so much? Well, Benzer's incredible experiment did more than just map a virus gene. It proved that a gene was a physical, tangible thing. It wasn't some abstract concept or an unbreakable bead, but a linear stretch of DNA with a real, mappable geography. This profound shift in thinking from an abstract unit to a physical structure is the absolute foundation for all of modern genetics, from cracking the genetic code all the way to sequencing the entire human genome. In the end, Benzer and his colleagues shattered one of the most fundamental concepts in their entire field. And it kind of makes you wonder, doesn't it? What beads on a string models are we holding onto today, just waiting for the next outrageous idea to come along and break them wide open?